Greetings. My name is Matt Banks. I'm a professor of anesthesiology and neuroscience at the University of Wisconsin in Madison. I want to thank the organizers of this meeting, and in particular Bruce MacIver, for inviting me to participate in this symposium. Today, I want to talk about changes in cortical functional connectivity that are associated with transitions between arousal states during sleep and anesthesia. The results I'm going to share with you today came out of a collaboration between my lab and that of Dr. Kirill Norsky at the University of Iowa, and were published recently in Neuroimage. The central motivating question for these experiments is, what changes in the brain when we lose and regain consciousness? A question we address in a variety of experimental settings. Today, I want to focus on a comparison between natural sleep and propofol anesthesia. There are a variety of dimensions over which we can compare sleep and anesthesia, and I've listed a few of them here. For some, such as whether or not the subject is arousable, and whether there is an underlying architecture to the experience, sleep and anesthesia clearly differ. In others, there is considerable overlap. For example, the involvement of subcortical sleep and arousal centers in the brain, the differential sensitivity of primary versus higher order cortex, and for some anesthetic agents, changes in delta power in the cortical electrophysiological signal. Interestingly, when it comes to measures of connectivity in the brain, there are divergent results. Some studies show decreases in connectivity, either global connectivity or more specific pathways, such as cortico-cortical feedback pathways. Other studies show the opposite result. Some reasons for the divergent results may be that different connectivity metrics were used in different studies, and importantly, the same subjects were not studied in the two conditions. So we wanted to investigate changes in functional connectivity in the same subjects during natural sleep and anesthesia. To do that, we obtained intracranial electrophysiological recordings from neurosurgical patients at the University of Iowa who are having electrodes implanted on and in their brains as part of their treatment for intractable epilepsy. These subjects have two surgeries, separated by about 2 weeks. In the first surgery, they have a couple hundred electrodes implanted over wide areas of the brain, including extensive coverage in temporal lobe and frontal lobe. These electrodes are used to determine the patient's seizure foci and to map particularly critical areas of the brain in preparation for the second surgery. In that surgery, the electrodes are removed and the seizure focus is resected to alleviate their epilepsy. Between the two surgeries, we recruit these subjects to participate in a variety of research studies, including those I'm presented here. Because most epilepsies involve the temporal lobe, which is also the location of auditory cortex, many of our research studies are focused on the auditory cortical hierarchy, and that is how I've color-coded the electrodes in these images. The dataset for this study was obtained from 5 subjects with roughly comparable coverage, extending from primary auditory cortex on Heschl's gyrus to secondary and tertiary auditory areas in temporal and parietal cortex, to higher order language processing areas in prefrontal cortex, as well as a variety of other regions. To investigate changes in functional connectivity during transitions between arousal states, we recorded resting state data from each subject during overnight sleep and during anesthesia. The paradigm for one subject is illustrated here. The sleep experiment is illustrated on the left, showing time on the horizontal axis, and the ratio of delta to beta power in one electrode, as well as the EMG signal on the vertical axis. Using standard sleep scoring methods, we labeled each 30-second segment of sleep data according to its associated sleep stage, wake, N1, N2, N3, and REM, color-coded here below the EMG signal. Because only 3 of the 5 subjects showed appreciable periods of N3, we'll focus our analysis mostly on N1, N2, and REM. The anesthesia experiment is illustrated on the right. It was performed in the OR just prior to the second surgery, as the subjects were gradually anesthetized with increasing doses of propofol. Here we're plotting 2 measures of arousal on the vertical axis, the bispectral index, an EEG-derived measure, and a standard assessment of awareness in the OR, the observer's assessment of arousal and sedation, the OAAS. We used the OAAS to determine whether subjects were sedated or unresponsive under anesthesia. Our hypothesis was that states N2 and N3 when we recorded it during sleep, during which dreaming is relatively low in frequency, and the state unresponsive under anesthesia represent states of reduced levels of consciousness, whereas wake and N1 and REM during the sleep experiment, when dreaming is relatively frequent, and the sedated state during the anesthesia experiment would be states of relatively high levels of consciousness. The metric of functional connectivity we chose is the weighted phase lag index, or WPLI, which is a measure of phase synchronization between two signals. It's calculated in the frequency domain, so we need to specify which frequency band we're interested in. Here, we're mostly concerned with the alpha band. It's symmetric and computed pairwise, so it includes both direct and indirect connectivity. It's designed to ignore zero phase lag differences, that is, simultaneous signals, as these likely reflect volume conduction. Importantly, it is unaffected by changes in power that occur in specific bands during arousal transitions during sleep and anesthesia. When we calculate the alpha WPLI for a specific chunk of data, we get an electrode by electrode connectivity or adjacency matrix, as shown here on the left. Here, the electrodes are sorted roughly hierarchically, with primary auditory cortex, HGPM, at the top left, and progressing to higher order auditory and prefrontal areas, sensory motor areas, and areas not directly related to auditory processing. Note the color scheme here is the same as was shown in the electrode coverage slide. To facilitate computation and account for differences in the number of electrodes in each cortical area in each subject, we reduce the dimensionality of the matrix by computing connectivity on an ROI by ROI basis. That's shown here in the center for the same data. Finally, an alternative representation of these ROI-based data is the chord connectivity plot on the right, where each ROI is a node on the circle, color-coded as above, and the strength of the connection is indicated by the thickness of the lines. For display purposes, these connectivity plots have been thresholded to show only the strongest 10% of the connections. This threshold was not applied for any of the quantitative analyses I'll be presenting. As I mentioned earlier, each time slice of data recorded during sleep and anesthesia has assigned to it a label corresponding to the simultaneously determined arousal state. Here, I'm showing adjacency matrices computed during an overnight sleep experiment, some associated with wake, some N1, N2, etc. We can summarize connectivity for each arousal state by averaging over these time slices. Other frequency bands did not exhibit this pattern, for example, delta and gamma shown here. So this summary plot supports our hypothesis that cortical connectivity changes upon transitions into states of reduced levels of consciousness, depicted here by transition across a boundary indicated by the vertical dashed line. However, this figure, as striking as it is pretty abstracted from the original data. It's averaged over time and over subjects. It's also only showing the 10% strongest connections. We'd like to show quantitatively that this transition boundary exists. That is, that the connectivity profiles overall are more similar between N2 and N1 than between wake and N1, or between unresponsive and sedated compared to sedated and wake in the anesthesia experiment. To do this, we employed two complementary analyses. First, we used a permutation analysis to determine the significance of changes we observed with the chord connectivity plots. Using the data for each time slice, we can compute the pairwise differences between adjacency matrices with different labels as a measure of how different connectivity is in one arousal state versus another. We do that by computing the norm of the difference matrix. I'm illustrating the difference between wake-sleep and N1, quantified as the average norm of the pairwise differences between all wake-sleep and N1 segments. We can then scramble the labels and compute null distributions to test significance. When we do that, we find that the difference between wake-sleep and N1 is significantly smaller than the difference between N1 and N2, shown in the top row. And the same is true for wake anesthesia versus sedated, and sedated versus unresponsive in the bottom row, indicating that there is indeed a transition in the connectivity profile upon crossing this boundary between arousal states of greater versus reduced consciousness. The second analysis we applied was a classifier analysis, again based on the adjacency matrices calculated for each time slice with its associated label. For each subject, we selected a subset of the wake-sleep and N2 time slices and trained a linear classifier to distinguish between the adjacency matrices for these two states, using a logistic regression output to yield a value of 1 for most wake-sleep-like and 0 for most N2-like. We then submitted the rest of the data, including the rest of the wake-sleep and N2 data, to see how the classifier distinguished the various sleep stages. In general, the classifier performance supported our hypothesis. It easily classified the rest of the wake-sleep and N2 data, indicating that we were not overfitting the model, and it classified N3 as being decidedly N2-like, and N1 and REM as being more wake-sleep-like. Furthermore, it easily classified the wake anesthesia and unresponsive data as being wake-sleep-like and N2-like respectively, indicating that there is an intrinsic similarity between alpha band WPLI connectivity profiles during sleep and anesthesia. For the sedated state, the result was more wake-sleep-like than not, but it clearly represented a transitional state, perhaps indicating fluctuating levels of arousal during propofol sedation. We also noted that the dominant connectivity shifted from the posterior to anterior parts of the brain during reduced levels of consciousness associated with sleep and anesthesia. That is, from temporal and parietal to frontal. This is reminiscent of the argument put forth a few years back by Melanie Bolie and colleagues about the location of the neural correlates of consciousness. Here, content-specific neural correlates of consciousness, or NCC, indicated in red, are defined as those areas directly contributing to phenomenal distinctions, for example, low-level visual features, faces, or places, within consciousness. The full NCC, outlined in orange, is constituted by the union of all the content-specific NCCs. The transition boundary that we observe is strikingly similar to the boundary between the full NCC and areas outside the full NCC. We quantified this shift by computing a measure of regional bias in connectivity, defined as the difference in mean connectivity between all ROIs in the back of the brain and the mean connectivity between all ROIs in the front of the brain. There was a clear shift in this regional bias with transitions into states of reduced consciousness, with dominant connectivity shifting from Bolie's full NCC to frontal areas in states of reduced levels of consciousness. In summary then, functional connectivity, as measured by alpha band WPLI, indexes arousal state. There is a similarity between cortical network organization during propofol anesthesia and sleep, and states of reduced consciousness, N2, N3, and unresponsive under anesthesia, are characterized by reduced posterior and enhanced anterior connectivity. Finally, I want to thank a remarkable collection of colleagues and support staff who helped with this project, as well as NIH and my department for funding. Thank you for your attention.
Hi, my name's Bruce MacIver. I'm a professor of neurophysiology in the Department of Anesthesia at Stanford University. Today I'm going to talk about nitrous oxide, also known as laughing gas. Some of you may have experienced it in dental procedures, tooth extraction, for example. It's commonly used in surgical anesthesia with a mixture of agents to produce analgesia, loss of consciousness, and relaxation. Now, it's a funny agent, and we'll get into that in a minute. But first, I want to thank my co-authors, TJ Shin from Seoul National University, who recorded the data, and Sarah Eagleman in my lab, who is an expert on digital analysis and helped a lot with the frequency domain measures and did virtually all of the complicated statistics used to process these signals. So nitrous oxide is weird because unlike other sedatives, it doesn't markedly slow and depress EEG signals. So other agents like Propofol, for example, produce large amplitude slow waves known as delta activity that is very similar to what you see in slow wave sleep. Nitrous doesn't do that at comparable levels of sedation compared to, say, Propofol. Also, none of the clinically used brain function monitors like the BIS or the SedLine are able to detect the effects of nitrous oxide, whereas they reliably detect sedation produced by other agents like volatile anesthetics, propofol, barbiturates, etc. So nitrous doesn't alter either the BIS or the SedLine analysis. So we're asking three questions today. Why don't brain monitors detect nitrous effects? Does nitrous do anything to the EEG signal? Maybe that's why the monitors don't work if there's no change in the EEG. And then I'm going to talk about a relatively new kind of analysis of EEG signals that was first developed by Stu Hameroff back in the late 80s. He was one of the first people to bring a computer into the operating room and record EEG signals of patients undergoing anesthesia. We've refined that technique. We can do the analysis faster, and we can display results in more clearly understandable ways. And we can quantitatively measure the effects of anesthetics, sedatives, hypnotics on EEG signals. And I'll show you how we do that in a little while. So to begin with, TJ got 15 volunteers and placed EEG electrodes on their scalps and administered nitrous oxide up to 50% in oxygen as the carrier gas, and then simply recorded the brain activity on multi-channel EEG recording equipment. Now, one of the first things you'll note is that nitrous can produce this highly synchronous activity across the whole brain. It starts in the frontal area and propagates backwards to the back of the brain. We're looking here at the frontal lead there versus the parietal lead on the opposite side of the brain and near the back of the brain. You can see these highly synchronous waveforms propagate backwards from the frontal cortex in continuous waves to the rear of the brain, the parietal areas. The frequency content is nearly identical across the frontal electrode compared to the parietal electrode. There's a lot of slow wave activity and this interesting alpha beta frequency rhythm between 10 and 15 hertz that comes and goes, waxes and wanes throughout the exposure period. And it's seen again both in frontal and parietal cortex. Now, in addition to variability within a subject with the signals coming and going and amplitudes changing, there were marked differences across the subjects. Different volunteers had much different EEG levels in the sedated, coming up to the 50% nitrous oxide, and following loss of response to various stimuli such as verbal commands, light touch, visual stimuli, eye tracking, etc. This would be at the deepest levels of nitrous at 50%. Some patients show a marked delta-like activity that's similar to the slow wave sleep patterns one might see, or the slow wave activity produced by other agents like propofol, except that it comes and goes much more inconsistently and quickly within a subject, even at the highest level studied, whereas propofol would produce a solid, steady level of slow wave delta activity. Some subjects, like B here, have quite a different pattern of activity. You can look just at the raw EEG traces. You've got the nice high amplitude slow wave activity here compared to a lot more fast activity. You can see that out here, and there's the alpha beta peak I was talking about earlier. Some patients have a prominent peak, other patients it's just barely even there. So a lot of variability within subjects and across subjects. That's probably why the BIS monitor and the SedLine monitor don't work very well with these signals, because they're changing in a way that kind of looks like what happens in an awake brain. A lot of variability and coming and going of different frequency bands, and a lot more higher frequency activity is preserved in some subjects. So that's partly why we're not seeing strong effects with the clinically used monitors. If we look across all the subjects, you can see there's hardly any change in any of the major EEG frequency bands from delta to higher frequency 40 Hertz gamma oscillations. So you look across any condition, sedated to the deeply sedated, loss of response level versus recovery, and there's very little change. A little bit of an increase in delta activity, depression in the theta band. Alpha and beta, as predicted, don't change much at all. So bottom line was there are no significant differences seen across any of the conditions at any of the frequencies. So again, this is why the monitors don't work so well. Now, I'm going to talk about this new analysis that Stu developed a number of years ago. The way it works, it's a nonlinear dynamic approach to the EEG signal. So we take the very same EEG signals and that we use to look at spectrograms and Fourier transforms, etc., that didn't produce much change in any of the frequency measures, but using a three-dimensional time-delayed embedding to produce attractor plots of the EEG signals, you can see these dot plots are the attractors coming from this EEG signal, this one's for that EEG signal, and this is for the recovery. Here we can see a distinct change if we measure the width and the length of these attractors. They're generally elliptical ellipsoids in three-dimensional space. Of course, we're looking only in 2 dimensions at these images, but this would be much like a rugby ball that shrinks down more like a football at the deepest level of nitrous exposure. And the width to length ratio decreases from 0.5 to 0.39 and then bounces back up on recovery. So clearly we're seeing changes across conditions with the chaos analysis. And if we look across all 15 subjects, we see statistically significant changes between sedated, loss of response, and recovery conditions. You notice across the population, we don't get quite as big a rebound in recovery as we saw in the previous subject, but clear recovery is seen. If we just look at each individual subject, you can see they all show this decreasing trend as we get to the loss of response stage with 50% nitrous oxide. And these all come bouncing back again on recovery, as shown before. I want you to pay attention to the huge amount of variability seen across subjects. These folks up here are virtually awake. The chaos analysis of their EEG shows a high level of symmetry, indicative of a high degree of complexity in the EEG signals. This complexity is generally attributed to carrying more information or indication of the brain doing a higher level of information processing. So with the nitrous oxide, we're getting a consistent loss of information processing capacity that correlates nicely with the deeper levels of sedation. If we were to normalize each patient, we could get highly significant differences between the whole population. So if everything was normalized to 100% of control, then this would be significant at the 0.001 or less level. So the chaos analysis is working pretty good, even on just raw EEG signals coming from patients or subjects that are clearly more aroused than others that are much more sleepy, looking at the lighter stage of sedation. Of course, in the awake subjects, they're all closer to 1, which is a sphere in 3 dimensions and indicates 360 degrees of freedom of information processing or complexity. So in conclusion, brain monitors don't do a good job of measuring nitrous oxide effects because the EEG shows only small and highly variable changes in the presence of this agent. Nitrous does produce a pronounced synchronization in the alpha-beta band in the majority of subjects, but varying degrees are seen across subjects. Some show, like the one I showed in the very first recording, shows highly synchronous alpha-beta activity. Others, it's there, but not a lot. The chaos analysis worked really well. We got highly significant changes across the 3 conditions we looked at, and it indicates that nitrous, like other sedative and hypnotics, is decreasing the complexity of the EEG signal, reflecting a loss of information, content, or processing in the brain. Thank you very much. Happy to take any questions.
Hi, my name is Heonsoo Lee. I'm a postdoctoral researcher from University of Michigan. Today I'm going to talk about spiking pattern of individual neurons under conscious and anesthetized conditions. Before talking about spike activity under anesthesia, let's talk about brain state first. We believed that anesthesia induces specific neural activity pattern of brain state which changes as a function of anesthetic concentration. For example, at very low dose anesthetic, we would see paradoxical excitation. In deeper anesthesia, we would see slower and high amplitude EEG or LP. At very high dose anesthesia, we would see burst suppression pattern. Likewise, most studies assume that brain state has a one-to-one correspondence with anesthetic concentration, and therefore they performed concentration-dependent approach. However, recent studies found different brain states in a fixed anesthetic concentration, and furthermore, they observed one brain state in different concentrations. Here, at fixed isoflurane concentration, the power spectrum can spontaneously fluctuate. Here in another study, the authors identified 9 distinct brain states based on EEG functional connectivity. And you can see that over this long time period, there are many transitions between different brain states. These findings suggest that local brain states spanning hundreds micrometers defined by spiking activities of neuronal population may also have a many-to-many correspondence with anesthetic concentration. Therefore, we identify local brain states under anesthesia and examine state-dependent changes of spiking patterns of individual neurons. This study will allow us to better understand neuronal basis of anesthetic-induced states of unconsciousness, and also many neuroscientific data is obtained under anesthetized condition. Therefore, this will also help interpret the neuroscientific data. So this is the experiment we performed. First, we implant multi-electrode array here on right primary visual cortex and record spike activity. We also gave light flash here as a visual stimulation. And this is the experimental protocol. We used the anesthetic desflurane and decreased its concentration in a stepwise manner. Here, the gray color represents recording time, and cyan and magenta represent spontaneous and stimulation session respectively. Today, due to limited time, I'm going to focus on spontaneous activity data only. From this recording, we could identify spiking activity of individual neurons. And based on their spike waveform, we classify them into white spiking, the red color, and narrow spiking, the blue color. The white spiking neurons correspond to putative excitatory neurons, and narrow spiking neurons are expected to be putative inhibitory neurons. The first goal of this study is to identify brain states based on spiking activity of neurons independent of anesthetic concentration. Therefore, we chose measures that can be applied to spike trains to quantify the well-known effects of anesthesia. The well-known effects are reduced spiking activity, temporal fragmentation of spikes, reduced neural complexity, burst suppression pattern in high-dose anesthesia. These were quantified, these spike measures, total number of spikes, mean spike rate, mean local variation, sample entropy, longest period below mean. I'm not going to explain the details of these measures now, but feel free to ask a question later. Here, the time course of the 5 spike measures were visualized along with the LFP spectrogram. You can see that the spike measures spontaneously changed even during the fixed anesthetic concentration. For example, at 6%, many measures changed abruptly. The spikes measures were fed as an input to the clustering algorithm. The method divided the dataset into 5 brain states, S1 to S5. The 5 states are here denoted by different colors. So let's look at the occurrence of the brain states in the data. This pie chart represents the relative frequency of the brain states in each anesthetic concentration. In general, S1 and S3 were mostly seen at 6%, S2 at 4%, S4 at 2%, S5 at 0%. Obviously, S5 represents wakefulness state. The important thing here is that the brain state and anesthetic concentration did not have one-to-one relationship. For instance, S1 was seen during 4% as well as at 6%. These are the representative LFP traits. LFP in S1 displayed burst suppression pattern. S2 and S4 revealed relatively high amplitude, slow activity as generally expected in anesthesia. In contrast, although S3 was mostly seen in 6% death brain, this showed relatively low amplitude and desynchronized LFP pattern, distinct from S1. The LFP pattern in S3 raised the question whether the arousal level may also be elaborated in S3 as it is in S5. Therefore, we measured EMG activity to follow the level of arousal. Although both S1 and S3 occurred mostly in 6% death brain, EMG of S3 was substantially higher than that of S1 or S2. Here is the statistical comparison. The 7 lines represent 7 animals. Statistically significant differences in the rescaled EMG were found for S3 versus S1 and S3 versus S5. Now we compare the 5 brain states in terms of both the number of emitted spikes and the average spike rate of individual neurons. The trace of mean SR spike rate and TNS total number of spikes deviated from each other, especially at 6% death brain. The TNS showed a pronounced decrease when the brain state transitioned from S3 to S1, whereas the mean spike rate remained the same. In S3, many neurons were inactive, even more than S1 and S2, but there were a few neurons with very high SR. Accordingly, the variation of SR across individual neurons was the highest in S3. The Gini coefficient of S3 was significantly larger than all the other 4 brain states. Next, we investigate the temporal dynamics of spike activity. Here, the black dots are rest plot. Vertical axis is for neuron index. As you can see here, spikes in S1, S2, and S4 were synchronously fragmented. On the other hand, S3 and S5 have asynchronous firing pattern. The figure below is about interspike intervals, ISI, of 7 representative neurons. The red color is for wide spiking neurons, and blue is for narrow spiking neurons. Notice that the vertical axis here represents ISI value, not the neuron index. The shape of ISI distribution was profoundly altered by the anesthetic. In wakefulness, the ISI distribution was unimodal, whereas in the other 4 brain states, it was bimodal or multimodal. This was partially due to the silent period in the spike activity. The large ISI value near 1000 milliseconds correspond to silent period that contribute to an upper peak in ISI histogram. In addition, some wide spiking neurons in S1 and S2 showed burst activity that was associated with very short ISI near at 10 milliseconds. In this slide, we focused more on the bursting activity of wide spiking neurons. This is an autocollogram of wide spiking neurons. As brain state changed from S5 to S1, the occurrence of short ISI was dramatically increased. In this analysis, we excluded the inactive or silent neurons with spike rate less than 1 Hz. Here, I compare the burst activity of 2 extreme cases, S1 versus S5. Each dot here represents a single neuron. Here, the burst ratio is defined as a ratio of the number of burst activity, that is, the number of spikes whose ISI is less than 10 milliseconds, to the total number of spikes. In S1, there are many inactive neurons, that is, neurons with spike rate less than 1 Hz, and the remaining active neurons have a high burst ratio. In S5, many inactive neurons became active, and burst ratio was decreased. Next, we examined a relationship between individual neurons and population activity. We calculated cross-correlation between spikes of a single neuron and spikes of a population. a substantial change in correlation was observed in both wide spiking and narrow spiking neurons. All pairwise comparisons were statistically significant for wide spiking neurons. High neuron-to-population coupling in anesthesia suggests an increase of shared information. However, because the entire population acts like a single neuron, the information content in the neural network should be extremely limited. This may explain why we are not conscious during slow oscillations. Now we measure transfer entropy, a measure of information transfer between individual neurons. Because spike itself is in part a result of a neuronal interaction, TE is expected to be positively correlated with spike rate. Here, x-axis is spike rate of receiving unit or neuron, and the y-axis is spike rate of sending neurons. And the dots represent individual neuron pairs. And the color of each dot represents the TE value, blue being low and red being high TE value. Because the spike rate is high in S5, there are many dots in the upper right areas of this panel. Because of this, the sum of TES, is highest in S5 compared to other 4 brain states. Neurons with SR spike rate less than 1 Hz were excluded from this figure. In contrast, we found an opposite result with the mean TE, TEM. The mean TE was lowest in S5. This is because for equally active neurons, TE is higher in anesthesia than in wakefulness. Let's look at the black square here. For example, the dots in black square here have more reddish color than the dots in here. Finally, we examined the distribution of TE. We calculated the Gini coefficient to quantify TE transfer entropy disparity. Gini coefficient of S5 was lower than all the other 4 states, suggesting that the dispersion of transfer entropy is lowest in S5. This and the result of the previous slide can be explained by the information diagram. Here, in each circle represents the amount of information. This is the information of xt plus 1, xt, and this is for yt. The yellow area here represents the shared information between xt plus 1 and yt given xt. This is the definition of transfer entropy. As explained before, anesthesia reduces SR, spike rate, and many neurons become inactive or silent. For the inactive neurons, the shared information is negligible. For active neurons, on the other hand, because they have bursting spike pattern and are synchronous with each other, the transfer entropy is also high. This explains why the total TE is low in anesthesia, while the mean TE and disparity are relatively higher than wakefulness state. So these are conclusions. The local cortical stage identified by spiking pattern displayed degeneracy in their relationship with the anesthetic concentration. This is consistent with the previous EEG studies. A previous unidentified paradoxical state, DS3, was found in high-dose anesthesia. The asynchronous firing pattern and high muscle activity suggest a possibility of transient awareness in this state. In general, under desperate anesthesia, some neurons became silent while others became bursty and synchronous with each other. Thank you very much for your attention.
My name is Shin from South Korea. I will talk about the changes of cognitive functions in Propofol-induced deep sedation. This is my short biography. And this is the introduction. My field is dentistry. The use of sedation is increasing in the field of dentistry. Drug-induced sedation is an effective adjunctive measure for dental treatment, especially for patients with high anxiety. Sedation is a continuum ranging from awake state to anesthetic state. Thus, sedation can be understood as a borderline state between awake and general anesthesia. Deep sedation is described by ASA as a drug-induced depression of consciousness during which patient cannot easily aroused or respond purposefully to repeated or painful stimulation. Understanding the mechanism of deep sedation could help in better understanding the process of the loss and recovery of consciousness. However, while the effect of anesthetic drugs upon cognitive function during perioperative period are quite well established, only a few studies exist regarding the changes in cognitive function during deep sedation. The audible paradigm is one of the ways to evaluate the brain's information processing, especially cognitive function for stimulus changes. The audible paradigm is an experimental method to observe the reaction when two types of different stimuli are applied irregularly. At this time, the standard stimuli refers to a stimulus that is frequently repeated, and the target stimuli refers to a distinct stimulus that is rare among the standards. When this external stimuli applied, information processing occurs in the brain, and the characteristic translocation generated accordingly. This is called event-related potential, ERP, and can be measured through the EEG experiment. ERP is triggered by brain activities to respond to external stimuli. With the utilization of audible paradigm, subjects are required to utilize cognitive function. This elicits specific type of ERP. P300 is one of the best documented ERP of this kind, elicited after a certain time period following the introduction of a stimulus. P300 is thought to be activated as a result of attention-dependent cognitive task. Therefore, changes in the P300 is expected to reflect changes in the level of consciousness and attention. In this study, Propofol was noted as a drug to induce sedation. Propofol is one of the most widely used drugs as an intravenous anesthetic used for general anesthesia as well as sedation. As a sedative agent, the use of Propofol is increasing for the purpose of endoscopic sedation as well as dental sedation. It exerts rapid onset and recovery profiles. Compared to midazolam, widely used as sedative agent, issue fast recovery. The pharmacology of Propofol has been extensively investigated. Therefore, we can titrate the effect of Propofol by using T-cell pump with utilizing Propofol PK/PD models, making titration easier. Unlike conscious sedation, deep sedation has several risks such as inadequate airway patency and ventilation. Thus, it is mostly utilized in setting where the titration of drugs effects are available. Recently, the application of Propofol deep sedation for dental procedures is increasing with its favorable properties. In the present study, we analyzed the changes in ERP, most notably P300, in an auditory passive audible paradigm in Propofol-induced deep sedation. We aimed to investigate how cortical and cognitive activity is altered in a borderline state between sedation and anesthesia. Because many anesthetic agents tend to target GABA receptor like Propofol, results produced by Propofol acting on this receptor can be expected to reflect the sedative process induced by other medication as well. This is material and method section. 20 volunteers ranged from the 20 to 40 years old were recruited. Each volunteer were provided with informed consent. Experimental methods were approved by IRB prior to conducting experiment on volunteers. Volunteers who had significant medical disease and laboratory abnormalities were excluded from the study. Each volunteer were instructed to fast for at least 8 hours before the experiment. Experimental protocol was as follows. First, baseline EEG was recorded for about 5 minutes. Each recording was performed on each subject and the computer-generated auditory stimuli. Two different auditory tone sequences were randomly delivered through the earphone for about 8 minutes. Propofol was administered to reach deep sedation. After that, two different auditory tone sequences were randomly delivered through earphone for about 8 minutes. After the discontinuation of Propofol, each recording continued until the subject reached the consciousness to the baseline levels. Each cap was selected to match the head circumferences of each volunteers. After setting the each cap 32 electrodes in the standard C2020 international placement were placed. One additional EMG channel was placed on the right outer canthals to remove muscle artifact. Each was recorded reference to the average EEG while keeping impedance at all electrodes below 5 kilo ohm. Each was sampled using custom-made software at a sampling rate 2048 Hz. Throughout the experiment, we utilized auditory audible paradigm. Two different auditory tone sequences were randomly delivered through earphone, 1000 Hz standard tone and 100-200 Hz infrequent distracted tone. The ratio of standard tone to distracted tone was 4. The auditory stimulation time of each tone was 50 milliseconds with a rise and fall time of 10 milliseconds. The time interval between each tone was 800 milliseconds. The auditory audible task consists of the mixture of 500 tones. With the use of MATLAB software, the trended function was used to remove each channel's linear trend in acquired EEG signals. The trended signals were filtered with a bandfast filter between 1 Hz and 30 Hz. The bandfast EEG signals were then corrected with epoch from 100 to 800 milliseconds from the sound onset. Next, starting 100 milliseconds prior to onset stimulus, the continuous signals with a 800 millisecond time window onset was epoched. The two-dimensional topographic map of scarf data field was generated with 40 millisecond windows to average out each channel's voltage in awake sedation stage. Each channel ratio, which had a statistical significance in all moments, were plotted using the Propofol function in EEG rep. The maximum and minimum EEG voltages between 250 and 500 milliseconds from the stimulus onset were obtained as a P300 peaks of each channels. The time between the awake sedation state, the peak voltage between each state, the peak voltage and voltage at the time of the anterior condition were compared using the paired t-test. In all statistical analysis, a p-value less than 0.5 was considered statistically significant. This is the picture taken in the experiment. This is the EEG device. The audible paradigm was generated by commercial software installed in these computers. Each was recorded with custom-made software. This is the result. In this study, we observed an increased potential in 300-400 millisecond ERP in P300, especially in FJet and CJet placement. It was observed P300 amplitude was higher under 1200 Hz tones compared to 1000 Hz signals. Notable reduction of the P300 under sedation was detected. Topographic distribution of ERP amplitude in the 320–360 millisecond latency range are shown in this slide. The left figure is the average ERP distribution in the arousal state. A cynetic shift of the curve indicates the areas with positive going ERPs. Peak amplitude can be observed in the central region in the awake state. The right figure are the topographic average ERP distribution in sedation state. The ERP amplitude showed significant reduction compared to the arousal state. The ERP amplitude differences between arousal and sedative state are shown as p-value distribution of statistical differences. Peak differences can be observed in the central area. Topographic distribution of ERP amplitude in 360 and 400 millisecond latency range are shown in this slide. The left figure are average ERP distribution in the arousal state. A cynetic shift of the curve indicates the areas of positive going ERPs. Peak amplitude can be observed in the central regions in the awake state. The right figures are topographic average ERP distribution in sedative state. The ERP amplitude showed significant reduction compared to the arousal state. ERP amplitude difference between arousal and sedative state are shown as p-value distribution of statistical differences. Peak differences can be observed in the central and peripheral area in this time range. Based on the above result, electrode Fc1, Fc2, C3, CZ from the central and parietal regions were selected to observe the temporal changes in ERP. This is the average time domain graph of Fc1 electrode in standard and target stimuli. Red lines in each graph represent ERP in the arousal state. Blue line represents ERP in sedative state. Statistically significant difference between each awake sedative state are shown in green boxes. Red dotted lines were used to enunciate P300 difference between arousal and sedative state. As shown in this figure, the amplitude was significantly decreased in sedative state. This is the average time domain graph of Fc2 electrode in the standard and target signals. We could see the same trend as the result of Fc1 channel we saw earlier. This is the average time domain graph of C3 electrode in the standard and target signals. We could see the same trend as the result of the frontal channels we saw earlier. In parietal channels, and the amplitude was significantly decreased in the sedative state. This is the average time domain graph of CZ electrode in standard and target signals. We could see the same trend as the result of the frontal channels we saw earlier. In parietal channels, the amplitude was significantly decreased in sedative state, as shown in this figure. P300 amplitude detected with an all-selected electrode showed statistically significant decrease in sedative state compared to awake state. This is the discussion of this study. In order to know what these changes in the P300 implication is necessary to understand the subcomponent that make up the P300. The P300 consists of two subcomponents, P3A and P3B. It is known that the P3A has a shorter latency, occurs mainly in the frontal area, and appears in response to rare new stimuli. P3B has a longer latency, mainly occurs in the parietal area, is known to appear in response to stimuli that require specific tasks to be performed. In this study, subjects were not subjected to specific tasks. Therefore, it can be seen that P3A is the main subcomponent that makes up the P300 from the previous result. What kind of information processing process the P300 reflect is explained by a theory called context updating theory. According to the theory, the brain creates a mental model, a hypothesis for stimuli based on previously accepted stimuli, and the P300 occurs when a new stimulus is recognized and the brain modifies or updates these mental models. Existing studies have interpreted the meaning of the P3A and P3B based on this context updating theory. In the case of the P3A, it is explained that it occurs in the process of recognizing and focusing on new stimuli that are different from before. P3B is described as occurring in the process of updating the memory and accessing memory store to compare this stimulus to the remembered stimulus. The result of this study can be interpreted based on the interpretation of existing studies on P300. As a result of the experiment in this study, the amplitude of P300 significantly decreased in Propofol-induced sedation. Considering the main subcomponent constituting P300 in this study is P3A, it can be seen that sedation using Propofol can weaken the cognitive process responsible for controlling concentration and discriminating between stimuli. In summary, it can be said Propofol sedation causes a decrease in P300 ERP, which reflects the weakening of the concentration process in sedative state. Thank you for attention.
Hi, my name is Grace Jang, and I am here to talk about ketamine's synaptic and neuronal effects. I worked on this project with my PI, Dr. Bruce MacIver from Stanford University. Ketamine is a dissociative anesthetic that has been recently identified to produce rapid-acting, long-lasting antidepressant effects. Unlike traditional antidepressants, ketamine is known for its actions on NMDA receptors, which is a type of glutamatergic receptor. However, ketamine also acts on a variety of other receptors and ion channels as well, including GABA receptors, acetylcholine receptors, dopamine and serotonin receptors, potassium channels, sodium channels, and calcium channels. Thus, the mechanisms of action behind ketamine's effects are not well understood at this time. We decided to investigate this by measuring ketamine's effects on synaptic transmission at NMDA and GABA-mediated synapses. We used electrophysiology recording techniques, and we recorded from the CA1 region of mouse hippocampal slices. We recorded population spikes as well as field excitatory postsynaptic potentials. These are our preliminary findings. At an antidepressant concentration of ketamine, ketamine was able to produce a small decrease in population spike amplitudes that was reversed during the latter half of drug application. Upon washout of ketamine, there was actually an increase in population spike amplitudes that was evident for up to 2 hours into recording post-washout. At an anesthetic concentration of ketamine, there was an acute depression in population spike amplitudes that was sustained for the duration of drug application. This effect was reversed upon washout, and the increase in population spike amplitudes was recorded for up to 2 hours into washout. We decided to see how long this increase in population spike amplitudes following washout of ketamine could last for, and we were able to record for up to 4 hours into washout. We decided to investigate the mechanisms that could be causing ketamine's effects, both acutely during drug application and long-term during washout. As you can see from this slide, there seems to be an increase in GABA-mediated inhibition in the presence of ketamine. So during baseline, the paired pulse inhibition ratio, which is calculated by the amplitude of the first population spike divided by the amplitude of the second population spike, was approximately at 0.8. However, after ketamine application, this ratio was increased to approximately almost 0.9, which indicates an increase in GABA-mediated inhibition in the presence of ketamine. We also tested ketamine on field excitatory postsynaptic potentials to determine whether or not ketamine's effect is at the synapses or via a postsynaptic mechanism. As you can see from this slide, field excitatory postsynaptic potentials were not as affected by ketamine as population spike amplitudes. In fact, during drug application, ketamine actually produced a small increase in field excitatory postsynaptic potential amplitudes, as opposed to the decrease seen during drug application for population spikes. However, ketamine was not able to produce a sustained increase in FEPSP amplitudes during washout like that seen for population spike amplitudes. Thus, we decided that potentially the mechanism that could be causing ketamine's effect is not at the synapses, but rather postsynaptically. We decided to confirm this using an IO curve that compares the population spike amplitudes before ketamine application to the EPSP slope before ketamine application, as well as that of population spike amplitudes after ketamine application and EPSP slopes after ketamine application. As you can see, there was an increase in this curve, which indicates an increase in postsynaptic excitability rather than synaptic transmission. This is because the FEPSP slope value remained the same before and after ketamine application, but for the same EPSP slope, there was a larger population spike amplitude that was produced post-ketamine application compared to pre-ketamine application. Because ketamine is known to act on NMDA receptors, we decided to test some NMDA receptor antagonists to see if they can replicate ketamine's effects. We first tested AP5, which is an NMDA receptor antagonist, to see if it can replicate ketamine's effects. As you can see from this slide, AP5 actually produced a similar decrease in population spike amplitudes like that produced by ketamine during drug application, and it was initially able to produce an increase in population spike amplitudes during washout, similar to that produced by ketamine. However, this increase in population spike amplitudes during washout was not sustained as long as that produced by ketamine, with population spike amplitudes decreasing back to baseline by approximately an hour and 50 minutes into washout. We also tested NMDA receptor ion channel blocker MK801 to see if it can replicate the effects of ketamine. As you can see from this slide, MK801 was actually able to replicate the acute and long-term effects of ketamine by producing a small depression in population spike amplitudes during drug application, as well as a sustained increase in population spike amplitudes during washout for 2 hours into washout. Because ketamine is also known to block potassium channels, we decided to test a global potassium channel blocker, tetraethylammonium, TEA, on population spike amplitudes. As you can see from this combined graph, TEA actually produced a much larger decrease in population spike amplitudes during drug application compared to that produced by ketamine. However, TEA upon washout was able to produce an increase in population spike amplitudes that was comparable to that produced by ketamine during washout. This increase was also sustained for up to 2 hours into washout. Here is a summary graph of all the drugs that we tested and compared with ketamine's effects. As you can see from this graph, the only drug that was able to produce a statistically significant decrease in population spike amplitudes during drug application was TEA, potassium channel blocker. However, during washout at both 60 and 120 minutes, ketamine at both concentrations, both antidepressant and anesthetic, as well as MK801 and TEA, were able to produce statistically significant increases in population spike amplitudes that was sustained for up to 120 minutes. Statistical significance was assessed using Wilcoxon rank test to compare experimental conditions with its respective controls. Statistical significance was established at p-value less than 0.05. In conclusion, ketamine produces an acute depression in population spike amplitudes via increase in GABA-mediated inhibition. Ketamine also produces a long-lasting increase in population spike amplitudes during washout due to an increase in neuronal excitability produced by a postsynaptic mechanism. APV was able to replicate ketamine's acute effects, while TEA, potassium channel blocker, was able to replicate ketamine's long-term effects. MK801, which is an NMDA receptor channel blocker, was able to replicate both ketamine's acute and long-term effects. Thus, the results indicate the involvement of NMDA receptor ion channel as well as potassium channel blocks in producing ketamine's long-lasting effects. And these long-lasting effects could be related to the long-lasting antidepressant effects produced by ketamine. Thank you.